Hello friends, this video on respiration in plants part 6 is brought to you by examfear.com. No more fear from exam. So what happens next in step 4? So in step 4, the starting material is going to be the product which was formed in step 3. So that is fructose 1,6-bisphosphate, right? So now the enzyme which will act here is aldolase, and the product which will be formed is, there are two products which will be formed: dihydroxyacetone phosphate and glyceraldehyde phosphate. So there are two products which will be formed in this step, right? So let us see what exactly happens here. So the starting material was fructose 1,6-bisphosphate. So there are 6 carbon and 2 phosphate groups, correct? So this is fructose 1,6-bisphosphate. Now this gets changed into 2 products, and one interesting fact is that these two are isomers of each other. So the first product and the second product, both of them have got the same molecular formula. It is just that their arrangements or their structures are different. So how is the structure? So if you talk, and each of them are a 3 carbon compound. So this was a 6 carbon compound. So basically it gets split into 2 3 carbon compounds. So let us see how the 2 3 carbon compounds are. So the first carbon compound would be somewhat like this, with 1 phosphate group. The second 3 carbon compound will again be somewhat like this, with 1 phosphate group. So basically it gets split like this. So now you have 2 3 carbon compounds with 1 phosphate group each. So you see here both of them have 1 phosphate group, right? And these two are isomers of each other. Both of them have the same molecular formula, that is C3H5O3P1. So this is the molecular formula for both of them, but the arrangements in both are different. So this is what happens, and it happens in presence of this enzyme called aldolase. So now you see finally in step 4, the 4 carbon compound, the 6 carbon compound got split into 2 3 carbon compound. So let's see what happens next. So in step 5, the starting material is going to be dihydroxyacetone phosphate, which was formed in step 4. Now in step 4, 2 products were formed. One was this one and the other one was glyceraldehyde phosphate. Now in step 5, what is done is basically this dihydroxyacetone phosphate is also converted into glyceraldehyde phosphate. Right? That means in step 4, 2 products were formed, 1 and 2. In step 5, 1 is also converted into 2. So that is what we are doing actually. So 2 will remain unchanged. 2 has nothing to do in step 5. So here an enzyme will be there called triose phosphate isomerase. So whenever you have this term isomerase, that means it will convert a compound into its isomer. Right? So now as I told, both 1 and 2 were isomers of each other. So now we want to convert 1 into 2. So that means we want to convert 1 into its isomer. So we use an enzyme isomerase. So triose phosphate isomerase is the enzyme which will be used here. And what will be the product? Product will be glyceraldehyde phosphate. Correct? So this is how it will be formed. So now the dihydroxyacetone phosphate, which will look somewhat like this. This gets converted into its isomer, which is again a 3 carbon atom with 1 phosphate group, just that the arrangements are different. So now in step 4, we already had 1 glyceraldehyde phosphate, which we have kept undisturbed. We did not do anything to that. Now here again, dihydroxyacetone is also converted into glyceraldehyde phosphate. So what is the net result of step 4 and 5? So the net result of step 4 and 5 is that we get 2 molecules of glyceraldehyde phosphate, right? So please understand this clearly. In step 4, you got one glyceraldehyde phosphate and one dihydroxyacetone phosphate. Both were isomers. Now what we did, we converted dihydroxyacetone phosphate also into glyceraldehyde phosphate. So now we have 2 glyceraldehyde phosphate. Clear? Okay, so we are halfway through glycolysis. Out of the total 10 steps, we have reached step 5. So at step 5, we have 2 molecules of 3 carbon compound glyceraldehyde phosphate. So now let us look at step 6. So here the starting material is of course glyceraldehyde phosphate, and step 6 is quite important and interesting as well. Now here we use an enzyme called triose phosphate dehydrogenase. Now we will see the speciality of dehydrogenase enzymes. Now here, this step in itself has got 2 sub-steps. So 2 things will be happening here. Let us see what is that. So here the product is 1,3-biphosphoglycerate. Okay, let's see how is that formed. Okay. Now here in step 6, we will be utilizing an oxidizing agent because oxidation will take place in this step. And what is that oxidizing agent? That is NAD+, that is nicotinamide adenine dinucleotide. So this is my oxidizing agent which will be utilized here. Now in the first step inside this, what will happen? This enzyme, that is triose phosphate dehydrogenase, it will transfer the hydrogen ion to NAD+ and it will form NADH. So this NAD+ will be converted into NADH plus H plus, and this will be done by the enzyme. And which is the enzyme? I am not writing the entire name of the enzyme because the name is quite big. It is triose phosphate dehydrogenase. So that is how it will oxidize this NAD plus. Now in the next step, what will happen? This same enzyme will add an inorganic phosphate from the cytosol to this oxidized glyceraldehyde phosphate. So the same enzyme will add one phosphate group. So glyceraldehyde phosphate. So glyceraldehyde phosphate is a 3 carbon compound with one phosphate group. So one inorganic phosphate will be added. So from where this inorganic phosphate will come? There will be so many phosphates present, the phosphate group present in the cytosol. So from there, one inorganic phosphate will be added to this compound to form the product. And what is the product? So product is 1,3-biphosphoglycerate. So that is again a 3 carbon compound, but here you have 2 phosphate group. That is why the name biphospho. So one phosphorus was already there, one more phosphorus will get added. That is why 1,3-biphosphoglycerate will be formed. Right? Now, this process will get repeated 2 times. Why 2 times? Because in step 5, 2 molecules of glyceraldehyde phosphate were produced, right? Now, for each molecule of glyceraldehyde phosphate, 1 molecule of 1,3-biphosphoglycerate will be formed, correct? So, since you had 2 molecules, therefore 2 molecules of 1,3-biphosphoglycerate will be formed. So let us, and also another important thing here is 2 molecules of NADH are also formed. So here if you see, this is also formed. Now these 2 steps will get repeated for each molecule of glyceraldehyde, right? So for first molecule, 1 NADH is formed. Again for the next molecule, 1 more NADH will be formed. So 2 molecules of NADH are also formed. Now that needs to be counted. That's because NADH are the energy-rich molecules. They are capable of producing, 1 molecule of NADH is capable of producing 3 molecules of ATP. So you can just imagine how energy-rich are they. So here in this step we are producing 2 molecules of NADH and 2 molecules of 1,3-biphosphoglycerate. So now as we go, thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.